So the second presentation, I'd like to give a very warm welcome to Dr. Peter Koeppel. He's written to the magazine several times, and I've always wanted him to come and talk, so finally we've got him here. Dr. Koeppel is a biochemist with a specialty in immunology, and he's been doing some pretty serious research in nucleotides. And from what I've read about it, it's a fascinating area, so I'm looking forward to his stuff. So, without further ado.
Thank you very much. I would like to thank a lot the organizer to invite me here, and I hope I can bring you something new about nucleotides. We are working in this company since more than 25 years on nucleotides, not only in the human field, but also in animals, mostly for animal nutrition. And here I would like to go mostly on the health and physiological effects of nucleotides, and then finally their effects on the performance. So our hypothesis was, is the right nucleotide blend missing in the normal diet of people? And in this presentation, I want to give you a little bit of information how important nucleotides are in food, and how, if you are looking at if your diets are really having the right amount of nucleotides for the people. Sorry. When we look on the nutrition, we see that nucleotides are more or less a forgotten nutrients, because all the food recommendations are based on recommendations on proteins, on fat, sugar, carbohydrates, and then on microingredients like vitamins, minerals, and trace elements. But nobody thinks about nucleotides. But nucleotides are really the basic for all the other nutrients, and we are considering our product as a micronutrient for which should also be included in our nutrition. Nucleotides, by the way, are in the category of Parnat's Commission regulations. Now, nucleotides have a major effect in the development of the body. And something quite interesting. When you look on the mother milk is extremely high in nucleotides. And all the feed, the formulation for baby food formulations were extremely low in nucleotides. And when the first babies were fed with this food, also baby food formulations, they found a lot of immature lymphocytes. They found a bad digestion, and they found a low development of the brain. So we were considering if nucleotides are not only good for babies, but also for adults. Now, you see, it is quite interesting when you check the nucleotides level in vegetables, fruits, meat, fish, and milk products, they are extremely low. And mostly they have a low bioavailability. And nucleotides are mostly found in higher concentration. I will show you some picture later, in everything that we don't eat, we throw away. For example, intestine, kidney, lung, everything is very high in nucleotides, but we don't eat it. But when you look, for example, on a predator, a lion, what he is eating first, he goes for the intestine. And nature makes always a sense why he's eating that. Because it's a lot of energy, it's a lot of basic nutrients, and we throw it away. And nucleotides in a high and balanced form, which has really a significant effect on health. And we call nucleotides as the best kept secret after the first 6 months, because during the first 6 months, you have enough nucleotides from mother or from milk supply. Now, when we go on the cell multiplication, on the cell, the body has a steady replacement of new cells. And you have always a degradation and formation of new cells. And the number of cells in the body is unbelievable, which are built every day. And nucleotides are the basic mostly for DNA and RNA, but nucleotides are also involved in other activities of the body. For example, for the whole metabolic activity. And therefore, I would like to give you first some background information about the nucleotides. Then the cells of every animal and every creature is more or less the same. The cells, they have a nucleus. It's the same in animals, in insects and humans. They contain DNA and can proliferate and divide, and they can produce more or less all the requirements for nucleotides. So even in the animal field, you have the same effects as we have here. Nucleotides are an integral part of the DNA, and a DNA is made up of 3 billion nucleotides, one single cell. And we have in the body a constant demand for new cell production. And the cell production always starts with the doubling of the DNA. And the doubling of the DNA needs, as I said before, these 3 billion nucleotides. And this has to be either made themselves or they have to come from somewhere else. But nucleotides are also involved in so-called protein synthesis, because you need messenger RNA, you need tRNA, and you need ribosomes for the protein synthesis, and these are all made from nucleotides. But nucleotides has other effects also. We don't know, most of the people don't know. They are acting as so-called second messenger. I come later on this, I will go in more detail later. They are part of the cofactors of some proteins, coenzymes, for example, some enzymes in the metabolism of fat, carbohydrates, and proteins. And they are the only way to transport the energy into the body. You have no other way than nucleotides. You have UTP, GTP, and ATP. And when there are not enough nucleotides, the energy transport in the body is gone. Now, just to give you a little bit more detailed background, also we have a nucleotide is made on a base, and the base can be either a purine, here we have adenosine, adenine, and guanine, or it can be a pyrimidine, and you see the purines, they have 2 rings, and the pyrimidines have 1 ring, and here you have cytosine, uracil, and thymine. And they are bound to a sugar. It can be either a ribose or a desoxyribose. And this part is called nucleoside. Also we speak about nucleobases, nucleosides, and then when you have here a phosphoric acid added, then it is a nucleotide monophosphate. And then we have nucleoside diphosphate and triphosphate. And the triphosphate is the energy transport in the body. But you have to please keep in mind a little bit that we have this differentiation between purines and pyrimidines. Now, the DNA is made up of a long chain of nucleotides, and it's always a binding between the sugar, you see here this sugar part, and the next phosphate. So you have a backbone here of sugar, phosphate, sugar, phosphate. And you have the side formation of the base, here for example guanine. And when we look, so here just an overview of the nucleotides again. Adenosine, when you have a nucleotide, or adenine, when it is a nucleobasis, guanosine, and then you have the thymidine, cytidine, and uridine. The nucleotides are involved in more or less all the activities of the body. There is nearly no process in the body which is not related to nucleotides. We have energy transport, we have the metabolic, we have catalytic effects, we have regulatory effects, and they are involved in structural functions. And therefore we have really a constant demand of new cell which needs nucleotides. And the nucleotide formation, also here you see, is the nucleotides are the basis of the DNA, which is the fingerprint of the body, or of the RNA. And you see the nucleotides some are the same, but some are different. In a DNA you have guanosine, cytosine, adenosine, and thymidine. Whereas in the RNA, messenger RNA and transcription, you have guanosine, like here. You have cytosine, like here, adenosine, like here. But here you have a different nucleotides, which is called uracil. So nucleotides are for both DNA and important for RNA also. Now, the cell multiplication always starts first with the doubling of the DNA. And you see you have in a starting cell, you have a double helix, and this starts to open and a new helix is formed. And as I told you before, you need 3 billion nucleotides. This means this part of the cell multiplication, where all the nucleotide has to be ready, is the longest part in the whole cell multiplication. Because the formation of, for example, the purines, you need 14 biochemical steps. And can you imagine you have 3 billion nucleotides you have to build within a certain time? So you see, only when you have enough nucleotides, and the right nucleotides, because you cannot here have another nucleotides. You need here, this is always this combination. You cannot replace one nucleotide by another. So all of them needs to be available in the right time. And I told you before here, this process takes roughly 8 hours, 8 to 12 hours. As soon as the DNA is here doubled, then the formation of 2 cells is only 20 minutes, because you have then here a double set of RNA, and they are slowly distributed on 2 levels. And you see you have the 2 sets, they separate, are drawn to this end of the cell, and finally the cells start to double, and then you have the 2 sister cells, which are absolutely identical to the first one. And that is the most important, because when you have here a mistake in here, either the cell is going to die, or it can be malfunction, or it can be even cancerous cell. So the right replication of the DNA is of most important. And the right DNA replication can only be done properly when the nucleotides are there in a sufficient amount. Now, where we get the nucleotides from? I told you we have, first of all, we have the de novo synthesis. The new synthesis of nucleotides is very limited. And I come here specifically later on, but it is also time consuming, as I told you before, but it's also energy consuming. It's a lot of energy going in, and you need a lot of basic materials to produce the nucleotides, mostly amino acids. The next way to get nucleotides is the so-called salvage pathway. The salvage pathway, in the salvage pathway, the nucleotides or the DNA from dying cells are recycled. But this recycling is only about 70–80%, not more. The rest is gone. And, and only those cells who die can be recovered. And then the nucleotides go in a pool, and they are then delivering the nucleosides for the next, let's say, cell multiplication. But here, a big part is also degraded, and you have then urate from the purines, or you have xanthosine and beta-amino isobutyrate. Now, I go a little bit here in the nutrition. I told you before that nucleotides are very important also in the food, because the body is not able to produce enough, so much nucleotides that it is self-sustaining. Now we have done quite a number of analyses of nucleotides, and we have here, for example, vegetables and fruits. And you see here, this is the percentage of nucleotides in a dry matter of the cell. You see, it is most of the, so the green one is the total nucleotides, the blue one are the pyrimidines, and the red one the purines. And you see, only broccoli, cauliflower has a certain number within the vegetables and fruits, which are enough, let's have a certain number of nucleotides. Most others have extremely low nucleotides. And I come later on this ratio between purines, pyrimidines and purines, and we find all the time that the purines are extremely high. And purines are, as I have said before, are degraded to uric acid. So people who eat a lot of can be then get gout. But the use really in the body is only on the level of the pyrimidines, because this is the limiting factor in the body. And you see here, this is on vegetables. Now I show you here, for example, in beef. And when you look, the scale is the same. And you see really a big part is in the intestine. Most of the nucleotides which you have is in the intestine. We have quite a good number here, for example, kidney, lungs, bone marrow, spinal cord is nearly nothing. But this is quite interesting. The ratio between purines and pyrimidines is nearly the same. But everywhere where you have very quick cell multiplication, also in eggs, you find a balance. Because the nucleotides which you need in the food should be balanced. But here, you see, this is extremely unbalanced. When you look, for example, in the heart, you have a lot of purines. This is clear because there you need a lot of ATP. The heart is always pumping, so a lot of ATP is there. So everywhere where you have a lot of, let's say, energy needed, you have a high number of purines. The same is here when we look further. Pork, absolute same picture. Again, a lot in small intestine, middle part, upper part, lower part. And then in the large intestine you have a little bit less. Brain, okay, it's not so much. But here, lung and kidney, you have again high amount. But who is eating this? Who? Today, we are throwing away. We say it is awful. My opinion is absolutely a wrong approach. We throw the most important part of the animal animals. We throw away without thinking what we are doing. Now here, this is just some mixed. You see here, for example, we have soya bean, extremely low. Mushroom is not bad, and also watercress. But for example, here butter or yogurt, even egg is extremely low. And also, and this is quite interesting, what we eat is fillet or muscle meat. And you see how much nucleotides it is in there. So we are really going, we do something wrong with our nutrition. And here again, I mean, when you look here, Marmite, yeast extract, when you use a normal yeast extract, again, you have a very high portion of purines and a low portion of pyrimidines. We have competition in the market, people saying we can use yeast extract for to give enough nucleotides. Clearly, you can do it, but you never have the right ratio of nucleotides as purines and pyrimidines. And here, just as a summary again, you see again here liver compared to all the vegetables. Liver is quite high. What is here? Liver sausage is not so good, but kidney, everything what we are not eating is really high in nucleotides. Now, something else, I was here speaking about the number and also the ratio. A number of nucleotides total and also the ratio between purines and pyrimidines. Now, something very important is also the availability, because nucleotides are normally not free, except of some, let's say, ATP and so on. Normally they are within bigger molecules. And these molecules are very well protected by proteins. Here, normally the nucleotides are in form of nucleoproteins. This means, for example, the DNA is surrounded by proteins, and they protect them against digestion, because here you have the whole information of the cell and of the body, and this needs to be protected. Same is true in the gut. So you have proteases, but these proteins are so formed that the digestion is very low. This means when we start here, you bring only about 5 to 15% of the nucleotides in the body, because nucleotides cannot be absorbed here. They cannot be absorbed as nucleic acid, as DNA or RNA. They need to be in form of nucleotides, or even lower in form of nucleosides, because here you have, in a nucleotide, you have a negative charge. And the gut lining cells are also negative charged. This means the absorption is relatively low. Here you have a neutral molecule, and it can easily be absorbed and taken up. And the best form to take it up is the nucleobases. And when we start here, when we start, for example, as nucleic acid, as pure RNA, or then we bring between 80 and 90% of the nucleotides in the body. So you see, we have several things which we have to consider. We have the amount of nucleotides, total amount. We have to consider the ratio between purines and pyrimidines, and we have to look for the bioavailability. And within this, when the nucleosides or nucleobases, they are in the lining cells, they are built to nucleotides, or they go into the bloodstream and are distributed in the body. So the first usage is always in the gut, and only afterwards you have the effect in the rest of the body. Now here is something about just a little bit, some numbers again. As I said before, the DNA is 3 billion nucleotides. So we need for every cell 3 billion. In a human being, every day, only blood cells are 2 billion are replaced. This means for humans, we need, this is 6 billion billions. How you say this? Nucleotides. I can even not express it myself. But this is only for cell multiplication. And you have to be aware, on one DNA, we have 1,000 RNA. This means the whole RNA part is even higher, is even 10 times higher than that. This means, you see here, we have different RNA products, also messenger RNA, tRNA, and so on. We have here the cyclic, also the nucleotides which act as a second messenger. We have here the nucleotides are as cofactors of enzymes. For example, coenzymes. We have in the body about 50 to 100 enzymes which are dependent on nucleotide cofactors. And last but not least is the energy in the body. As we see here, ATP and GTP. This is also the reason why we have more purines than pyrimidines in the body, in the meat. Now here, just a little bit an overview of how our body, how quick the body is replaced. That you are every 7 years a new person, except of the brain. About 5 to 7 years, your body is totally new. Because you see here you have the bone cells also, it's quite long, but muscle cells, they maximum go 10 years, 10, 15 years, something like that. Fat cells are only 8 years. Lymphocytes, some of the lymphocytes are about a year old, can get a lifespan of a year. But you see, for example, liver, let's say every year, you have more or less a new liver. You have red blood cells, it's only 120 days. Platelets, only 10 days. And now we come here to what happens in the intestine. The colon has about 3 to 4 days. And you know how big the colon is? Also, when you look at the human body, the body is about 2 square meters, the surface. The lung is about 20 square meters. And the intestine is between 200 and 400 square meters. It's a football field. Just remember, every 3 days, all the cells are replaced there. In the small intestine, even 2 to 4 days, stomach 2 days, and blood neutrophils, also the defending cells, the important defending cells, also 1 to 2 days, 1 to 5 days. So you see how quick we are going, and we are replacing. And all these processes are depending on the nucleotides. And nobody, I'm quite sure most of you are not really looking what the nucleotide content is in your food, and your food for your patients. Now here, I repeat, because this number is for me striking. A day, this number of nucleotides to build in the body or to require the body is unbelievable for me. I cannot imagine for me. Now, we were referring mostly to cell multiplication. But the importance of nucleotides are also for example, in metabolic activities, in the transfer of energy, hormone signal. Here, very important tissue repair. And now I was very interested about the presentation of Justin before, because gut integrity is really a major issue. And when you see how quick gut is replacing cells, you see what this means. Now, something very important also, and here I come then later on oxygen radicals, is DNA repair. Because this is whatever happens on the DNA, when you have there a damage, a cell is going to die. And then here, finally, this effect on protein synthesis. Now here, the whole transport of energy is, and here you have the so-called adenosine triphosphate, and you see you have 3 phosphate. And by releasing this last phosphate, you release also the energy into the body. But this is really the only way to have energy in the body, together with these 2, GTP and UTP. Also everything, wound healing, muscle work, everything is depending on enough energy in the body. Now, something which most of the people are not aware, when you have a hormone, the hormone comes down to a cellular level. And here you have the cell wall the wall of a cell in the body. Now, when you have a first messenger hormone, it gets some on the cell surface on a receptor. How is now this information going inside to trigger, for example, enzyme activity or DNA activity? You have here a whole chain of effects. At the end, here this ATP is transferred in a cyclic AMP or cyclic GMP. And you see you have hormones, they have an activation of this, and some hormones, other hormones, they break down this. But always here you need a second messenger. This means nucleotides are also very important in the hormone activities of the body. Otherwise, the hormones cannot really bring the activity into the cell, in an activity in the cell, because hormones always release a DNA, also protein synthesis. The next step, what I wanted to tell you is about wound healing. What happens in wound healing? When you have a wound, a small cut, you have a lot of damaged cell. And the first step in this is that macrophages come into this wound and they start to clean the wound. And they eat. And when they overeat, then you get a very heavy chronic inflammation. But it's really the cleaning of the wound through the macrophages is extremely important. And we have done in our own labs trials on mice, and we looked when we feed a normal diet or a diet with nucleotides, and we looked when we stimulate the immune system, because something I have to say, our product is not a stimulation. It's just to give a fuel for something to do. In this case, when we gave the animals nucleotides, we have 4 times more macrophages compared to the control group. This means the wound healing is much quicker, and as soon as the wound is cleaned, the same macrophages releases substances to activate the surrounding cells to proliferate, also to make new cells. It means the first step here is really an immune reaction. And then for the wound healing, again, you need really a considerable amount of of energy. So wound healing is extremely important. Then here, the same, I have not so beautiful picture as Justin had before, but this was a trial which we did in animals. And you see, we fed the same animals for 3 weeks, either a normal diet or with nucleotides. And quite interesting is you see the villi length here was 25% higher. We had a mucosa area which was much better, because when you look here in the corner, you see this mucosa layer. And this is very good formed, whereas when you look here, you have a lot of open space. And when you have this open space, you can have bacteria which go into the villi and cause some inflammation. And for example, in IBS, there is new studies showing that micro lesions are the main reason for IBS types of symptoms. So when you can, let's say repair the whole, or make the repair process quicker, then you have less of a problem, because the whole cell multiplication is only down here in the crypt, in the gut. And then the cell, they migrate along the villi up to the top, and at the top they are shivered away. This means when you have not enough nucleotides, the villi will go shorter. Only when you have nucleotides in amount which is needed, then the villi first of all get bigger, digestion is better, and you have also a better integrity of the gut. And something quite interesting is, here we have also measured the enzyme activities in the gut, and we have found that more enzymes are produced. And this is again because I come later on the protein synthesis. To measure this effect, we have done a trial with people with irritable bowel syndrome. This was a trial here in East London University. We had here 37 people with the diagnosed. And the trial was so that we had a control group and a trial group. And they were 6 weeks, they get either the control diet or with the nucleotides. And then we have a crossover, but a washout for 4 weeks and crossover. Also it was the same type of trial as you do in a pharmacological trial. Also we had the same people were once the test group and once the placebo group. And they have every 7 days they have to fill in a questionnaire. And we assessed abdominal pain, diarrhea, urgency, incomplete evacuation, flatulence, and so on. And we had this product, because this is a special product which we have made for gut health. And here you see the effects which we have seen. We have quite a good effect also on placebo. But you see here, for example, abdominal pain highly significant improved. Diarrhea was improved. Urgency was improved. And incomplete evacuation was improved. These three, and these all were significant. These three were not significant, but you see that there is also an effect here. And most probably, we did the whole trial not long enough, because this was the performance. You see the baseline, the symptoms were more or less on the same level. With the placebo, we see a difference, but also over the time, we did not see an improvement. Only here, with the nucleotides, we started nearly on the same level here, but over the time, we found an improvement. And this is very important because the gut also needs time to really fully recover. And at the end, this was the big difference between the placebo and the trial group. Now, it was quite interesting, the comments of the researcher. Also, we had really seen a benefit over the placebo with our product. The variation within the patient, within the symptoms was lower. We have seen really an effect, a big effect on pain, abdominal pain, feeling of urgency, and feeling of incomplete evacuation. This was really very much pronounced. Then we had, because of the small number of people, it was only 37 people, we had this last part we have not improved. We think that with a bigger amount of people we would have also a significant there, and mostly also when we could use it longer. But to keep people in a trial was always difficult, because we had a lady who phoned every day, every patient, if they have taken the product. Only in this way you can really keep them going. But you see the general effect, all were in the same direction. And this product is a nucleotide formula, which we made specifically. And we made the nucleotides so that the purines were low and the pyrimidines were high. Because we wanted then to, and this you can only do when you do with purified nucleotides. You cannot take any food to balance the feed. That is not possible. As you have seen from the trial or from the test results in food, it is we didn't find anything which will have more pyrimidines than purines. So the only way to do it is to make pure preparation of the single nucleotides and then mixing together in a certain formula. And we have combined it with some vitamins and some amino acids, and this was, but the biggest effect, also the real effect, was only here. Now, something quite interesting also, because we are going now in the intestinal flora, it is that some of the gut flora are not able to produce nucleotides. And this is, for example, some of the bifidobacteria or some of lactobacilli are not able to grow without nucleotides. This means they are depending on nucleotides. And when we have fed, we did mostly in animals because there you can make sections. When we fed nucleotides to animals, we always found an increase of bifidobacteria. And we have also seen those bacteria who are immune stimulating in the gut. For example, the so-called Apopondium group, we have also found higher amount there. And so here you see that other effects you need also, other ingredients you need also, but the most important are, for example, here you see adenine, xanthine, uracil. To really grow these bacteria. Now, I said before also that the integrity of the DNA is most important for a cell survival. And here we looked what happens when we release an oxidative stress, what happens on the level of DNA. And you see here we have the first group here, in the control group, the whole energy came out, or let's say 95% of the energy came out of starch. Here we have replaced starch by linseed oil. And linseed oil is quite rich on polyunsaturated fatty acids. And we have here 20% fatty acids, here only 3%. And this releases a so-called oxygen radical burst. In this group we have done absolutely the same as here, but here we have the nucleotides. And you see here the energy is same, but the proportion coming out of fat is here 5%, here 30, and here 21 against 3. And what we did, we took lymphocytes, we isolated the DNA and put it on an electrophoresis. a DNA of a cell is about 1 meter long. When you put it on an electrophoresis, it doesn't move. So all the stain, and then you stain these, the nucleotides. All the stain is here when you have an intact DNA. When the DNA is cut in parts, you have a tail, and therefore it is called comet test, because it looks like a comet, and you can measure how much you find here and how much you find in the tail. And you see in the beginning, also before the test, we have in all 3 groups, we have about 96% intact DNA. Now, after 21 days, either on a control diet, you see here, there is still 96% intact DNA. But here, when we have the oil group, it was 15% damaged. 15% of the lymphocytes were damaged. When we have the same group, but we add nucleotides, we come on the 6% and the 4% damaged as we had before. This means when you have enough nucleotides, the body has the ability to repair the DNA. But nucleotides needs to be there, because when this repair mechanism stops somewhere, the cell is lost. So by giving nucleotides, we can also overcome, let's say, stress released, for example, of oxygen radicals. And interesting is that every sport, every effort releases a lot of oxygen radicals. A wound healing or an infection, the first defense in the body, cells of the immune system releases oxygen radicals to kill the body, to kill the bacteria, not the body, sorry. To kill the bacteria. But now you have the bacteria, but with this product, the body cell around this infection can be protected or can be repaired. The bacteria, because they have a single DNA, it's not double, they cannot repair because they have no counterpart. So the bacteria can be destroyed, whereas the cell of the body can be, let's say, repaired and saved. And you see here, this is a typical picture of this comet test. You see here, everything is nearly here in the front, and here you have the tail. You see, this is all damaged DNA, and when we put the nucleotides, we found again everything here in front of the DNA. Now, protein synthesis. I have to hurry up. You see, protein synthesis starts always in the DNA. You need the DNA, but then the DNA opens and gives a way to produce a so-called messenger RNA. The messenger RNA with the information for a protein lines up on the ribosome, and then these tRNA, they bring the single amino acids to the place, and they are then connected by the energy which you get from the nucleotides, from the so ATP and GTP. This means for a protein synthesis you need 5 different nucleotide molecules. And nobody thinks about that. A lot of people, they take a lot of amino acids, proteins, without considering that nucleotides would be the first step, because this is the first step here in the whole protein synthesis. Sorry. Now, we were speaking about the body is able to produce nucleotides. Now, there are exceptions. The bone marrow-derived cells, also all lymphocytes, all macrophages, all red blood cells, are not able to produce nucleotides. They need it either from the liver, and when you have, for example, a disease, normally liver function is going down, or by the food. So here we have always seen a very, very big effect on nucleotides. The intestinal cells, they are able to produce nucleotides, but their own production is by far not able to cover the needs of the replacement for the cells. Then as I showed before, intestinal flora, also bifidobacteria, and here, this is quite interesting, is brain cells. And there are trials ongoing, for example, for memory effects in older people. And they have seen that that the memory effect, also memory is much better when you have, when people have fed with nucleotides. I jump because I'm running out of. Here, just some trials which we have done in immunological. Also, we see a higher resistance against microbial challenge. We see more interleukin-2. We see much more antibody trial production here, and the number of leukocytes and neutrophils are after an infection is much higher. And here, this was a trial which we did in Edinburgh, and we looked at people with the cold. Also, people were coming to the university with a cold, and we looked here on the painful sinuses. And you see in the control group, after they come to the university, the symptoms went up and only then went down. When we had these nucleotides in here, after 5 days, the whole problem was gone. We have also looked what happens when we have sport people in sport, and we measured here the antibodies in the HEA antibodies in the mouse. We have seen after a high effort, the antibody in the mouse goes 90% down. It's amazing, 90%. And we have seen here, we have done 2 trials here. This was here in UK, and this was in a moderate endurance effect, high endurance effect. And we see here we have 10% more, and here 20% more of antibodies. Here you see the effect again. But why is this? Because we have also looked we measured also the cortisol response. And we found, again, in these same trials, also here in the same trials, that the build-up of cortisol was here 10% less. And in this new trial, which was just finished in America at the moment, we have 25% less of cortisol build-up. Here you see, again, this was the trial in America. The experimental group and the control placebo group was in the same, and here you have this difference. Now, the interesting part here is that normally we measure only once the amount of cortisol, and here we measured before the exercise. It was a really intense exercise. Directly after the exercise, then 15, 30, 60, and so on. And you see the same people were again placebo and trial group. And you see in the placebo group, it goes up, significant up, goes down and come up again. When we have nucleotides, you find here more or less all the same on the same level. We have, by the way, we did same trials also in animals. And now here, this is quite interesting also. We have looked on the sprint time. Here, this was this is in Liverpool. And this trial was so that they have 6 times 35 meters in maximum speed. And the average in the placebo group here was 7.06 seconds. In the group with nucleotides is 6.6. And we have here a much improved peak from force. And this is again here, again the same trial as before. This is before, immediately after, and then 24 hours, 48 hours, and 72 hours. And you see in the control group, we have a breakdown of the force. Only after 72 hours they recover, whereas the nucleotide group more or less remained on the same level. So people can directly, after high exercise, they can go back into the training. And we have seen this very often with people, with marathon runners. They told me normally they need about 7 days to recover. And with the nucleotides, they're between 1 and 2 days afterwards, they were again in the training. So we call nucleotides as a conditional essential nutrient. And we think sooner or later we are in a status where nucleotides are really essential. Mostly in certain diseases, when you have a limited intake of nutrients, heavy exercise, or elevated stress. This is not only in sport. We have seen the same effect also with people who have mental stress. The same effect we have seen there. And the own production of nucleotides, they require a lot of time, as we have seen, 8 hours, which they need normally. And we have done trials where we looked under antibody building in animals. We have seen the antibody titer were doubled in a control group within 8 hours. And when we have very high amount of nucleotides, it was in 2 hours were doubled. So the antibody titers were also the whole nucleotides needs a lot of time and energy, and we help the body really to save energy and to do the right thing in the right time. So with this I would like to stop. Otherwise I speak the whole afternoon. Thank you very much.
Thanks very much, Peter.
No problem.
I'm going to start eating liver and taking some nucleotides.